want to the order of things.
Okay, so we're presentation official since this has started to be recorded. So thank you everyone for coming. It's very nice to see you here. I know it's a very busy time of the year. We're also very, very grateful for our graduating seniors. We've had really the extraordinary privilege to share several years with you, in some cases 4 years with you, and to see you grow and develop. And it's just as a professor to have that opportunity to meet a very young student, general biology, or maybe a sophomore little class, and then see them grow, go on to do internships, and take all these great classes. And in many cases, they wind up doing research in a laboratory and then go on to bigger and better things. And then we might be able to see you a few years down the road. And it's one of the great experiences we have, one of the most rewarding parts of the job. So we're very grateful for you. Now, we're going to start. So one thing I should mention is that we have an outline of the program and the nominating statements on this QR code. So there's some papers on the sides of the bench. So if you want to scan that so you can read the statements or see what we're doing, you feel free to do that. And so the order will be, we were fortunate enough to have 5 students nominated for the Graduating Biology Student of the Year Award. So we will have the professors that nominated them come up and tell us about why they nominated those students. And so we'll go through that first. Then we have an oral presentation by Matt Bielda, and we will do that. And then after that's done, we'll go outside. There's going to be some refreshments and some beverages, and we will have poster presentations by another 5 students. There's also a poster with some biological art that Dr. Richardson facilitated for us with one of our students. So that's the order of things. Okay, so first off, I'd like to ask Dr. Kozlowski to come up and introduce our first nominee.
It'll save time if we both log on at the same time. Thank you, Windsor. Hi, everyone. For those who don't know me, I'm Dr. Kozlowski, and I was very fortunate to nominate Jessica Martinez. I met Jessica, so like many of the seniors, freshman year was online, and after your freshman year, for those who came during freshman year, people were starting to come back to campus, and Jessica got to come back to campus as a rising STEM scholar and got to actually experience research with the cohort model. I was one of the mentors. She got to be in my group, and although she was a little shy and hesitant, she turned out to be a fantastic student and decided to come and work in my lab, which she's been working in for the last 3 years. Jessica's a real go-getter because I gave her a bunch of really bad brain tissue to try and work with. And because of that, she had to really implement a lot of new techniques that we didn't do in the lab, figure stuff out on her own. And I'm very pleased to say she's not presenting today because she has 2 more brain slices to analyze. And they will be analyzed before she graduates. So we may actually have a graph before the end of the year. And those of you involved in research know that sometimes it takes that long to get a graph. But aside from this, she has received undergraduate summer research program awards, as well as has been an LSAM Louise Stokes Minority Participation Scholar for all her years here at DePaul as well. And she's going to stay at DePaul and be part of the master's entry into nursing program. So congratulations. This is
yeah, there you go.
get the program in the background.
And I'm, yeah, so thank you.
Thank you.
And I don't mean to dine and ditch, but I gotta go to the neuro.
Okay, so I will go next and introduce a couple of students. And unlike Dr. Kozlowski, I will have to read my statement. So the first student I'd like to introduce is Hema Landa. So unfortunately, Hema couldn't be here today, but I nominated Hema for the Graduating Student of the Year award. So Hema is a great student with that did very well academically in classes. She's passionate about biology. She joined my lab in autumn quarter of 2022, and she was also a student in the Rising STEM Scholars program, although I didn't work with that program that year. And she worked with me on several projects, including a project surveying skeletal variation in freshwater fish along elevational gradients in the Andes. And she's also worked on barcoding freshwater fishes from Western Ecuador, which is something we've been doing for years and hopefully getting close to the end of. Hema was also a member of the Rising STEM Scholars Program, which I already said. And finally, Hema obtained a funded summer research position in 2023, working with Dr. Ovji Chaudhuri at Stanford University studying intestinal organoid morphogenesis. Such advanced research, I can't even pronounce it. And Hema was actually admitted to the postbac program at Stanford University. So she's going back there and in the fall. So clearly has a very bright future. And then the other student that I'd like to talk about is here, and she'll be presenting a poster in the poster session, and that is Allison Ritter. So Allison is another student with a strong academic record, and Allison's one of the most passionate students I've met in terms of ecology and evolution. She's a very strong interest in that field. She also joined my lab in fall 2022. I remember she took the molecular methods and ecology and evolution course that she took, and you approached me and you said, okay, all the other students in the class seem to be in your lab. Can I also join? Something like that. And so I said, of course, because she was doing great in the lab. And I can tell she's just a fantastic student. She's enthusiastic, very engaged, great at the bench, skilled at doing the work that we do with DNA sequencing. And she obviously works very well in group settings. She's also worked on a couple of different projects. One she'll be presenting about it's actually a global collaboration led by researchers at the University of Bern, surveying stickleback populations from throughout the world, throughout the Northern Hemisphere for these chromosomal inversions that are adaptively important. So we contributed samples from Alaska, and Allison can tell you all about the results and very interesting patterns that we were not expecting. And then she's also, like a lot of the students in the lab, worked on the DNA barcoding component. We're barcoding freshwater fishes in Western Ecuador using 3 different genes, and moving into doing that with a new type of technology called nanopore sequencing. She also last summer assisted a graduate student. She got funding to assist one of the graduate students in the lab with collecting invasive round goby on the shore of Lake Michigan. And I should say, Allison's extraordinary fisher person. She was, we put the rods right in and she was getting them right and left. Much more quickly than I was, or graduate student should go unnamed. Graduate student did great too. So Allison is also pursuing postbac programs and applying to those, and is interested in applying to graduate programs in ecology and evolution in the future. So Allison, why don't you come up here for the picture opportunity? My phone is still active. Congratulations.
Thank you. Get out of the image, yes.
With that, I will introduce Dr. LaMontagne, who will talk about the last two nominees.
Okay, so I've got two students that I nominated. So the first is Dominic Radcliffe Hines. I can make you stand. So everyone can stare at you while I talk about you. That's fun.
Yeah, that's great.
Yeah, perfect. All right, Dominic joined my lab in fall of 2022. And I highly recommended him for the graduating biology student of the year. Dominic came to DePaul after completing a degree in theater. So that's a cool background. I had the top GPA of the 2024 graduating class in biology. Dominic joined the lab working on a task of separating the seeds of several different tree species from one another and other organic matter. This is like compelling work, but it's super important, right? Like this is how this stuff happens. It has to be manual stuff. So yeah, to support a long-term project on the study of tree reproduction, which is a tedious task that requires attention to detail, and it just needs to be done. So I thank you for all of those hours. Dominic has also been doing mentored research in biology courses with me, focusing on testing hypotheses about synchrony and reproduction between two species of co-occurring trees, and he's gonna have a poster on that work. So these are white spruce and balsam fir, and this project focused on data analysis. Dominic has been using and developing his coding skills to graph and analyze these data. And outside of the lab in summer 2023, he had an NSF RAU experience at the University of Tennessee, Chattanooga in mathematical biology. He's presented a research at the CSH Science Showcase at the Midwest Ecology and Evolution Conference, Chicago area undergraduate symposium as well, and will be starting a PhD program as well. So. And over to you for this one.
Yes. Thank you.
And then finally, Matt. And Clapper. And Matt was nominated by both myself and Dr. Hill and this award. And you get the award. Now I get to say nice things about you. That I highly recommend Matt Balta for the graduating biology student of the year. So Matt is a high achieving student among the highest GPAs of the graduating seniors. And I've always been very impressed. So you're positive in the lab, engaged in meetings, you're organized, and have strong initiative to make processes more efficient using technology tools. And has strengths in communicating research findings. So Matt joined the lab in spring of 2023 and participated in the CSH Master's Undergraduate Scholarly Engagement, so the MUSE program, with Adriana Yoder over there. Also did a URAP and independent study courses. So in addition to assisting with field work in the Chicagoland region, Matt used a strong background in computers and coding. So he did was a Bachelor of Science in, was it nuclear engineering?
It was nuclear
Yeah, I thought I had that right. Prior to transferring to DePaul. And he used GIS software first to analyze data on red-headed woodpecker nest site locations with proximity to water bodies. But then there were these thousands of photos that he will tell you about, so I won't steal everything. And it was like, okay, help process these and figure out, is there a woodpecker in these pictures or not? Which could be a very manual process. And Matt was like, there's gotta be a better way to do this. And using computers to help guide, identify when there's a red-headed woodpecker in there or not. So that was all Matt. So everything he's gonna talk about was his initiative to do that. So that's really cool. And there were thousands of these photos. And then more recently, Matt has been using machine learning to develop a workflow to automatically count cones on photos. And so we have a long-term data set. Again, he's gonna talk about that a little bit, where we take photos and then someone counts them using ImageJ manually, and there's gotta be a better way to do it. It's kind of Matt's idea. So, and again, we have thousands of trees and dozens of years of data now. And Matt has presented his research at the Science Showcase and the Midwest Ecology and Evolution Conference. So congratulations on graduating Senior of the Year.
Thank you so much. And Matt, no, don't sit down. Still have more work to do. But yeah, Matt is our graduating student of the year. So congratulations. With that, he's going to tell us about his research.
You can stand.
The floor is yours.
Yeah, so I did want to take some time today to talk to you all about the research I've been doing with Dr. Lemontaine in processing these large image data sets with computer vision. So Dr. Lemontaine set me up perfectly for this. Large image data sets are essentially just a huge collection of images that convey data visually, and so it's not a convenient way to numerically extract it. And then she did mention we have these two data sets in our lab here that we work with. First of all, the red-headed woodpecker cavity study that Addie is working on. And so this took place over the 3 months of the last winter. We had 24 cameras out in the field taking photos over those 3 months. And so as you can imagine, we accumulated hundreds of thousands of photos, I think, overall. And then the second project is Dr. Lemontaine's long-term cone counting project. This has been ongoing since 2012 up to the present. And so again, these photos have accumulated over years and years, and we're left with thousands of them. Yeah, the traditional way to use these photos is to go through manually and process them by hand, and that comes with a lot of disadvantages. Obviously, it's very time consuming when you have thousands of photos. Even if you can do 1 photo per second, 100,000 photos, 1 photo per second, that's still hours and hours and hours of work. So you might want to pay somebody to do that, but that can be very expensive, even if you're paying undergrads. And it creates inconsistency among the reviewers, right? If you have multiple people doing it, people's brains work differently, the patterns they see are different, and so you might have people that notice a woodpecker in one image but not another image, or vice versa. Or they're counting the cones and they might have a different method and miss 1 or 2 here and there, and so this can create inconsistency. And then finally, there's just this turnaround time that exists where you have your images and you'd like to be able to use them, do statistical tests on them, but you can't because you have to go through by hand and get the numbers out of the images. And so my objective with these 2 kind of projects was to determine a way to automate them, right? Make it faster, either aid or completely expedite the process of annotating the images so that we can focus on kind of the more interesting and important aspects of these studies. And so what is computer vision? And this is kind of a broad catch-all term. It just refers to any sort of algorithm that can extract information from image or video data. And so I'm sure all of you are very familiar with the tech that's being used in like smartphones now, assisted driving technology in cars. Here we have some medical imaging examples. And it's seen a lot of advance recently, past 10 years in particular, with things like machine learning really taking off. This field is growing very quickly. So I do want to play a quick little game with everybody here. So I'm going to show you an image from a camera trap, and I would like you all to look for the woodpecker. I'll give you about 10 seconds here. If you see it, you can raise your hand. Close to the front. All right, if you've missed it, or you're unsure, I'll reveal it here. So it's up there. And for those of you that do or did see it, I imagine the thing that drew your eye to it was the distinctive red head of the woodpecker. You can see it kind of stands out in the background there. And just like your brain uses that to pick up on unique things in an image, that is the technique I attempted to use to process our images with computer vision as well. So I do want to get a bit into how I did that. And so the first step in that was something called gamma correction. And so that is just adjusting the brightness values on a pixel per pixel basis of an image based upon how far away they are from kind of the brightness standard. And so we use a log scale to do that so that the darker or further away from the standard the pixels are, the more they get adjusted. And so if you, I'm not sure if it's actually coming in super well on the projector here, but you can kind of see that this image is a bit more easier to read. You can see some definition in the bark there compared to the left side image. And that's just to make it so that going forward we have an easier time looking for things. The second step is color masking, and this is where the looking for the color really happens. This is the core. And so we have an image of a woodpecker here, and then I've blown up the woodpecker for ease of seeing, and then over here is the corresponding mask. And so all we're doing is we're taking the regions of the image that fall within that hue and saturation range that matches with the woodpecker's head, and isolating them and making everything else in the image black. But that leaves a lot of other stuff as well over here, right? We have our noise that's coming from the dead leaves on the side. And we don't want that, that confuses the program. And so we can apply a bit of noise reduction, which allows us to remove a lot of the extraneous material while keeping our original actual subject. And so what we do with this is erosion, which every white pixel in the image that's bordering a black pixel is removed, right? So that shrinks every blob by 1 pixel, essentially. And then we do a dilation, which is the opposite of that. And so that brings it back up, but that creates a bottleneck where the smallest regions just get removed completely and they don't get redilated back up. And so that's how most of that material over there ends up being removed. So then the next step from there would be actually treating these blobs as blobs and determining their location in the image. So they become key points, which are just the coordinates of each blob. And then we try to isolate which key points are significant. And we do that by looking at how close they are to the other key points in the image, which allows us to kind of distinguish between a woodpecker and noise, right? If it's noise, it's going to be near other noise typically, whereas the woodpeckers tend to be on dead trees or in the air, and so they're not going to be among the dead leaves that create this interference. And so if they're significantly isolated, we can overlay them back over the original image and come up with this. You can see here, we have found the woodpecker. And so this seems to work pretty well in ideal conditions. And by ideal, I mean well-lit images that have a nice high contrast range, and the head of the woodpecker is visible, right? That's, if we're looking for the red, there's no red because it's like sticking its head in a cavity or something. It's just not going to work. And so I found that on kind of our best image sets from some of our cameras that happen to have better lighting and stuff, we had about a 40% positive ID rate, and so that means that other 60% was false positives. So locations in the image that weren't actually woodpeckers, but the algorithm identified as woodpeckers. But that's still cutting down on a lot of images to go through. But the issue is the performance suffers quite a bit when the images are like this, right? They're very low contrast, washed out, and it makes it difficult for even the human eye to see anything. And so obviously it's more difficult for the computer to use those images as well. And so our success rate on those is very low. But I do want to change gears a bit here and talk about the cone counting project. So this is just an illustration of essentially how many cones you can really see on one tree. You can kind of pick them out individually on this one, but then they've all been counted by a student in Dr. Lemontaigne's lab previously over here. And so there are hundreds on this one tree. And so this is, again, a time-consuming process to go through and count these all by hand. And so my goal was to use something called image segmentation to, again, try to speed this process up. And so image segmentation works off of a similar principle as the color mask, but it has a little bit more subtlety, a little more nuance to it, because it doesn't only just look at the raw color of each pixel, but it actually looks at how each pixel compares to the pixels around it as well. So that allows you to look at sharp change between adjacent pixels and determine things like edges in the image. And that means that we can do a better job identifying distinct shapes among similar colored things, because obviously a spruce tree with the cones does not have distinct red features that we can pick out very easily. And so that makes it a more robust system. But as you can see, the mask, again, has a lot of noise going on, and a lot of the overlapping cones tend to become one large blob. And so I tried I tried to break that up a bit by applying a water shedding algorithm, which just splits blobs into smaller blobs based upon how the water would flow through. But as you can see, we don't end up with cone shapes when we do that, right? We end up with sort of these little continents. And counting these up and just treating them as individual cones gets a number that's around 20% or so of the, or within 20% or so of the correct value in the tests I've run. But it seems like it's not finding cones all the time. It's just making up cones to make up for cones that it misses. And so I've kind of decided this particular approach is not worth really trying to pursue through the future. There we go. And so I do want to mention a few of the challenges I think that these field ecology studies face using computer vision compared to most other fields like medical imaging, where you maybe a more controlled environment. So first of all, there's a lot of variance between where you're taking your images. You don't have the ability necessarily to put the camera in the exact same spot every time. You're just putting it on a tree somewhere, hoping you get a good angle. And so you end up with a lot of kind of unique cases on a camera-to-camera basis, and that can make it difficult to standardize what you're doing. Additionally, the images themselves are very complex, right? You look at a tree has branches, each one of those branches has branches and leaves and stuff, and so it creates a very kind of messy image compared to something like a car that you would see where the lines are very clean and distinct, and you can block out what goes where easily. And then finally, over here, and this is probably the biggest issue, is the lighting inconsistency. Even if you have a site, among that one site, you have a lot of inconsistency in how the photos look. And so the pixels in this one are very blue, very bright white in this one, and that can create a lot of difficulty using any sort of color-based approach to analyzing images, even at the same exact site. However, I do think that there are some potential solutions to this as we go forward. And so most recently, I've been working on using machine learning, specifically convolutional neural networks, to try and take a more, I guess, modern approach to this problem. And so machine learning and neural networks essentially function off the idea of very similar to human brain, where you just put input in and connections are formed without us really having any impact on it. It just happens and then it puts out output. And so what you do is you train these algorithms by feeding them known data sets. And then once you've trained it, you can feed it an unknown data set and hopefully get good results. The reason why I think this is finally something that's worth pursuing, and maybe not 5-10 years ago, is that obviously computing power just gets cheaper every year. And we have a lot of resources now, like AWS or Google Cloud or something, to just get a hold of high power servers without having to have like a university license or anything like that. And then coming with that is the resources that are around, right? YouTube tutorials, blogs, academic papers. There's all kinds of material out now that didn't exist 10 years ago surrounding using machine learning in this sort of application. And then finally, and this is the biggest one that I'm excited about, is that both of these projects at this point have a large base of accumulated data that has already been annotated, right? So that's our training set for these algorithms. And so we have all the pieces we need at this point to really do this. And we might not have even, 6 months ago before the woodpecker pictures were counted. And so finally, the specific approaches I'd like to take are utilizing something called a UNet for the cone counting project. And that's given its name by the shape here. That's just the architecture of the program. And what that does is it compares the images to the masks. And so the masks in this case would be the post-counting image data sets with the white or red dot that you saw earlier. Comparing those two and trying to train it to know where the cones are in the image. And then for the red-headed woodpecker project, it would be a simpler neural network known as a regression. And that just takes one image with a value attached and then looks for what in the image causes it to have that value. So it just looks for common patterns, either in this case, presence or absence of a woodpecker. And then you'd be able to go through with a novel image set and look for woodpeckers. Before I go to questions, I do want to thank a few folks. First of all, Dr. Lamontagne for guiding me through all of this and meeting with me every week, just helping me think through things, as well as providing obviously the cone counting data set going back 12 years She's used the same camera the entire time taking the photos, so it's very consistent, perfect for this sort of thing. I'd also like to thank Addie for allowing me to use her red-headed woodpecker images, as well as providing a lot of input and guidance on just like, presenting things and running the project and stuff like that. She's been very helpful. And then finally, I'd like to thank Dr. First from the College of Computing and Digital Media for meeting with me and providing some input, particularly on the neural network side of things, and really encouraging me to move in that direction and making it a little more, accessible, I think, mentally. So yeah, thank you guys so much for listening to me. I'd love to answer any questions you've got.
Is it hard to tell this woodpecker from other ones?
Yeah, so this one is pretty distinct when you see it with the naked eye. Most other woodpeckers that are about the same size have, like there's the red-bellied woodpecker. So that doesn't actually have the same exact coloration. It's got more mottled stuff on its wings and stuff. For the program, I would imagine, yes, very hard to distinguish between, right? I think it would identify any woodpecker in the image. Fortunately, we're looking at cavities we know to be red-headed woodpecker cavities, and so it's much more likely to be a red-headed woodpecker than any other sort of woodpecker. Yes.
So, her talk. I really enjoyed the talk and then the use of the technology. Just but one question I had was in the situations where you have the camera taking basically static for long periods of time and at the same angle, and there's issues with the light. Is there any use to maybe putting a model in the frame that's the color you want? So if you have if you want that tone of red on the woodpecker, what if you put like a red color in there, and so there's a positive there. Say, okay, find something that's like that in this lighting.
That's an excellent idea. That would certainly be something that'd be worth trying out, I think. We had set up the cameras by the time I started working on this, and so I didn't really have the opportunity to do anything like that. But if this was something going forward where we had the opportunity to put out more cameras, I would definitely love to try that, because I think that sounds like a very easy way to kind of calibrate what you're looking for, right? Sounds like a great idea. Yes.
Great talk, man, and congratulations. So one of the issues that you mentioned was the false positive rates. Did you ever have any issues with like false negative rates where because my assumption is that the red color is based on some carotenoid pigmentation.
Okay.
And so if those birds are just like not getting a good diet, then there's going to be less red in their plumage. And so was there ever instances where the birds were there, but because they just weren't red enough, you couldn't pick them up?
Yeah, certainly. We have missed woodpeckers in situations where even if their head was out of the shot somehow, if it was tucked behind a branch or in a cavity. I'm not sure if we had any that were just off color or not, specimen wise, but certainly when the coloration of the entire image is skewed, that messes up. So I imagine if there were any birds with, less than the most vibrant red, maybe something more dull or empty, then it wouldn't show up as well. Yes.
Is there any sexual dimorphism between the colors of the woodpeckers between the two genders, and then could that affect the data sets?
Not in this particular species. That would certainly be something, if you wanted to apply this more broadly to other bird species, then you would have to really look out for that and either have two separate models or maybe even try to train one to look for both. But for these ones, we got lucky. Red-headed woodpeckers look very similar between the genders. So.
Have you had any thoughts about sharing this sort of information, sharing these sort of data sets with other people who are also doing these sort of ecological, surveys?
Yeah, I would love to share the kind of stuff I've worked through. The data sets don't technically belong to me, so that'd be more of a question for Addie and Dr. Lumatain. But at least the methods-wise, there's a really nice culture of kind of open source work for this sort of stuff. I used a program called OpenCV that provides a Python library to work with. And so that was, a huge factor of me being able to do this at all, is that there is this collaborative atmosphere. And so love to be able to feed into that.
One last question, not related to the presentation itself, but just maybe words of wisdom now that you're, one of the seniors and graduate student of the year. Do you have any advice if there's a younger student in the room about how they can make the most about from their college experience?
Yeah, I guess I can. I mean, this is gonna sound very kind of corny, right? But just look for the opportunities and try to do as many of them as possible. Like, don't overextend yourself, but I would read the bio class every week that Sarah sends out and just look for interesting things in that. That's, I think, how I found out about Addie's looking for an undergraduate to work with on her Muse project, and that's how I got into Dr. Lemontaine's lab and stuff. And so even before that, I saw an email that Dr. Sparks had sent out about his South Carolina trip, and that's kind of what made me want to do research at all. And so just trying lots of things, I guess, was, I think, the biggest factor.
Okay, well, thank you. We can give another round of applause to all the students who have been nominated for the Student of the Year. And maybe one more round of applause for all the graduating seniors. So, congratulations. With that, we have the poster presentations now, and there'll be some treats outside. And so thank you everyone for coming, and enjoy yourselves and have some food.